Hello, my dear student. Welcome to Pawan Wagh Academy. Myself Dhanvantari Deshmukh. Today, again we are discussing about reproduction in lower and higher animals. From this chapter, we have completed with asexual reproduction. We have seen that there are two types of reproduction. First one is asexual and second one is sexual type of reproduction. When we have seen asexual type of reproduction, that time we have learned different methods, that is gemelle formation, budding, that we have seen. We have seen different phases, we have seen breeders, and now today we are starting with sexual type of reproduction. Now, how the sexual type of reproduction is going to take place? We are taking the example of human reproduction here. Again, yesterday only I told you that what is meant by reproduction. Reproduction means to reproduce something. And if there is reproduction of something, it will require some energy. And again, the process of reproduction will occur by step-by-step events. And if it is occurring by step-by-step event, that means what happens? It has to complete according to serially. And if it is completing serially, then it has to complete first step and then only it is able to walk towards the next step. Now, we will start with first step of human reproduction. Now, here, first step is gametogenesis. This is the first step of human reproduction. Here, in this step, see, gameto means what? Gametes. Which are the gametes here? There are two different types of gametes. One is male gamete and other is female gamete. Okay, male gametes are known as sperms and female gamete is known as ova or egg. These are the gametes, male gametes as well as female gametes. Male gametes are sperms and female gametes are egg or ova. Now, what are gametes that you got to know? But what is happening with these gametes now in this gametogenesis process? Genesis means what happens? Genesis, the words genesis means formation. Genesis means formation of something. And here, formation of gametes is taking place. And the process of formation of gamete is known as gametogenesis. Gametogenesis is the process of formation of gametes. You got clear with this term? Gametogenesis. Gametes means there are two types of gametes, male gametes and female gametes. Male gametes are sperms, female gametes are egg. We are calling it as egg or the ova. And their formation, the process of formation of these gametes is known as gametogenesis. Okay? And again, the formation of gametes, the formation of male gamete, the process of formation of male gamete is known as spermatogenesis. Spermatogenesis. And the formation of the process of formation of female gamete is known as oogenesis. Okay. Both of these processes are going on. Where these processes are going on? The process of spermatogenesis is going on inside the male gonad, and the process of oogenesis will occur inside female gonad. Okay, now here you have cleared with how gametes are forming and by which process they are forming and in which area they are forming. Okay, this is known as gametogenesis. This is the initial step of human reproduction. And how humans are reproducing? They are reproducing sexually. We can say this reproduction as a sexual reproduction. Now, the second step in human reproduction is insemination. What is the second step? Insemination. Now, what is mean by this insemination? Insemination means introduction of semen into the reproductive tract of female. That is known as insemination. Introduction of semen. From where the semen comes? Semen comes from this male, from male gonads. From this male gonads, what comes? Semen comes, and that semen is given out into female gonad. And this process is known as insemination. The process of insemination. It takes place after the process of formation of gametes. Okay, and again here, see, there is the difference between gametes and gonads. Both of them are different from each other. Gametes are different and gonads are different. Gametes are the cells. Gametes means what? These are the cells, and these cells, the process of formation of these cells is taking place inside these gonads. Okay, and now after gametogenesis comes insemination, and after insemination what comes? Internal fertilization. The third process is internal fertilization. Now what happens in this internal fertilization? See, what happens here in internal fertilization? What we have seen, first of all, gametes we have seen. After that, the process of spermatogenesis and oogenesis will occur. And the third process is what? Insemination. This is the process of insemination. What is this process? This is the process of insemination. And in this process of insemination, the semen which contains sperms is given out into the female gonad. And after this process, after insemination, the process of internal fertilization occurs. Now what happens here? Inside the female gonad, what happens? The egg is present, and when the sperm, millions of sperms travels, and one of them fertilizes the egg. Now what is the process of fertilization? This process means what? At which state we can say that fertilization has taken place. See here, these are the sperms and it is a egg. And now from here millions of sperms are coming, and this one sperm, okay, this sperm, wait a minute, this one sperm when touches when this one sperm touches, see here, when this one sperm touches the egg membrane, at that time what happens? It touches the egg membrane and that sperm gets entry inside the egg membrane, not sperm. The head of sperm will enter inside the egg membrane. And what that head contains? It contains nucleus. And now actual fertilization process means what? When the sperm gets fused with this egg, we can say that the process of fertilization has occurred. And what will be the result of process of fertilization? After fertilization, what will happen? After fertilization, zygote formation will take place. Fertilization is a process and the result of process of fertilization is zygote formation. Yesterday I told you in detail how the zygote is formed. Again I will tell you today. Same. The sperm gets entry inside the egg and after that in the next step what happens? That sperm has given the nucleus inside that egg and that egg has its own nucleus also. So that's why now in this one egg there is presence of two nucleus. One nucleus, we are saying this nucleus or we are calling this nucleus as male pronucleus. Male pronucleus, because it had came from the head of sperm. Which nucleus we are calling it as a male pronucleus? The nucleus which came from the head of sperm which we are calling as a male pronucleus. And the nucleus which was there only, which is the nucleus of this egg or the ova, that nucleus is known as female pronucleus. Okay. Now, when both of these nucleus gets bound together and they gets fused, and in the next step, what happens? What happens here? The fusion of nucleus will take place, and they will form only one nucleus. Here, one nucleus is formed now after fusion of both of these nucleus. And now here we are able to say that it is a zygote. Got it? Again, in this process, in the deeper region, what happens? See here, male pronucleus. It has a set of chromosome, and again female pronucleus. It is again having a set of chromosome. And here male pronucleus is in an N condition and female pronucleus is also in a N condition, means haploid. Only one set of chromosome is here. 23 pairs of chromosomes are present in male pronucleus and 23 pairs of chromosomes are present in female pronucleus. And when they fuse together and the zygote is formed, and now this zygote we can call it as a 2N condition, means here are two sets of chromosome. That means 46 pairs of chromosomes are present inside the zygote. Okay, before that, before fusion of this female and male pronucleus, it was haploid in condition, but after fusion it becomes diploid in condition. This is the main thing or important thing in the human reproduction. Without this, nothing will go on in the right direction. Now, after zygote has formed, what happened next? After that, embryogenesis will take place. Now, what is embryogenesis? Embryogenesis means This is the embryo. Okay, means embryo. And this embryo now, okay, again here, it is a embryo, and here what happens, the formation of embryo will take place. Genesis means formation, and what thing is forming here? The genesis of what is going on here? The genesis means formation of embryo is taking place in this place. Now, in embryogenesis, zygote has formed, and after that, the embryo will be formed. Now, the next stage is gestation. What is that step? Gestation. Next step of human reproduction is gestation. Here, again we will clear with embryogenesis. The embryo will form. But how the embryo will form? Again we will see that process in detail. We have seen up to the stage of zygote formation. Now, after formation of zygote, what happens with that zygote? Now that zygote, it will, see here, now after formation of zygote, when we will call it as an embryo. After that, this zygote will travel to the next direction, but here the cleavage will take place. After that, the process of cleavage will take place. Now, what is meant by cleavage? What is meant by cleavage formation? Now that one cell, this is one cell. Now, after formation of zygote, it had become one cell. And now, this one cell is getting divided into two cells. Okay. And we are calling it as a two-cell stage. 1 and 2. And like this, what happens? The process of cleavage will occur till the formation of 64 stage. And again, the division of this cell will go on. After 2 cells, they will form 4 cell. After 4 cell, they will form 8 cell, 8 to 16, and 16 to 32, and 32 up to the 64 cell stage. After the 64, they have formed, and in between, what is going on again? The stages are known as blastula. After this, the blastula will form, and after the formation of blastula, gastrula will form, and after formation of gastrula, what happens? Morphogenesis will take place. And after morphogenesis, organogenesis will occur. And now, at the step where gastrula has formed, the embryo is implanted into the uterus. And after implantation, the process of morphogenesis and organogenesis will take place, and it will grow and develop inside the embedded layer of endometrium. And this endometrium is a layer present on the uterus. It is a thin layer, and in that the width of endometrium is more, and as the width of endometrium is more, this developed gastrula will again go and it will get implanted inside that endometrium layer. Now after that, what happens here? Sixth process, it is the period of gestation. Now, what is mean by gestation? Gestation is the period where we can again say it is a time between conception and parturition. Gestation is the period between conception and parturition. That period is known as gestation. And here what happens with that fetus? The growth and development of fetus will occur during this gestation period. Okay. Now the last step of human reproduction is parturition, and this step, the step of parturition is followed by lactation. How? Parturition means delivering out the baby from the uterus. But after this parturition, what happens? The process of lactation will also take place through the mammary glands. In this way, these are the steps of human reproduction, and it is a serial process. It is occurring by serial events: gametogenesis, insemination, internal fertilization, after that zygote formation, after that embryogenesis, after embryogenesis, what happens? Implantation will take place, and after that gestation will occur, and the last step of human reproduction is parturition, and after parturition the process of lactation will take place. In this way, we have seen, or we have discussed, the steps in human reproduction, or how the human reproduction occurs in the serial events. Now, first event in human reproduction was gametogenesis. That's why we are starting with gametogenesis. Now, what is gametogenesis? I told you before only that formation of gametes is known as gametogenesis. And this process of gametogenesis will take place in sexually reproducing organisms only, because they are reproducing with the help of gametes only. Now, this process again takes place in two ways. Just now I told you again same thing. Female gametes are ovum and male gametes are sperms. And how they are forming, how the process of gametogenesis is taking place. The process of gametogenesis occurs by they are forming with the help of primordial germ cells. Again, these cells, primordial germ cells, they are very important in gametogenesis because they are providing the base for the formation of gametes. These are the cells from which the gametes are originating, and where these primordial cells are present, they are present in the gonads. If the process of gametogenesis, that is spermatogenesis, is taking place, then at that time the primordial germ cells are present in testes, and according to that in the female gonads also. Now we will start with process of spermatogenesis. Here the formation of sperm is taking place. That's why we are saying the process as a spermatogenesis, the process of spermatogenesis. Now, here the formation of sperm is taking place, but in which part the formation of sperm is taking place? This process, the process of spermatogenesis, occurs in testes. Okay, and these testes, they are the part of male gonads. Now, in this testes, what happens? Again, there is a tubular structure. We are calling it as a seminiferous tubules. See, seminiferous tubules are there. This is the tubular structure where seminiferous tubules are present, and in that area the process of spermatogenesis is going on. And that seminiferous tubules, see now again, where the process of spermatogenesis is going on, it is going on in the testes. And where are the testes present? They are the part of male gonads. And again, in that area, seminiferous tubules are present, and this seminiferous tubules, at its lining, what is present? Germinal epithelium cell is present, and this germinal epithelium cell will undergo the process of spermatogenesis, and they are helping for formation of sperm. Got it? Means who had an important role here? Germinal epithelium cell. And this germinal epithelium cell, they are present at the lining of this seminiferous tubule. And where the seminiferous tubules are present, they are a part of male gonad, and they are present inside the testes region. Now, whenever the process of spermatogenesis is going on, that time again one thing is happening, that is, here in between germinal epithelium cells, which are at the lining of seminiferous tubule, these germinal epithelium cells, what they are doing? In between germinal epithelium cells, what is present? Nurse cells are present. We are calling them as a Sertoli cells also. And what will be the function of this Sertoli cells or nurse cells? Why we are calling it as a nurse? What nurses are providing us? They are these Sertoli cells. They are providing nourishment to the developing sperms. That's why they are known as nurse cells. They are providing nourishment, and because of that, the sperms are alive. In this way, what we have seen in this slide, here the spermatogenesis, the process of spermatogenesis is going on. Where it is going on? In the testes region. And testes region, these are the male gonads. And these gonads, again, that means these testes, they are a part, or they have a part known as seminiferous tubule. Seminiferous tubule. And this seminiferous tubule, again, this seminiferous tubule, it is lined by germinal epithelial cells. Germinal epithelial cells. And these cells, in between these cells, who is present? In between these cells, again, nurse cells are present. We are calling it as a Sertoli cells. Sertoli cells. And what will be the function of this Sertoli cell? These Sertoli cells, they are providing nourishment to this developing sperms. Got it? Now this is the initial stage of development of sperms. Now there are different phases while developing the sperm, and we are calling it as a phases of spermatogenesis. While forming of sperm, there are different phases, or the formation of sperm occurs according to different phases. Which are those phases? Here the first phase is multiplication phase. Second is growth phase, and third is maturation phase. These are the three phases of development of sperm. We will discuss these phases one by one. Now this is the first phase. It is known as multiplication phase. multiplication, means something is there and that something is going to multiply. And here what happens? Do you know what will be the difference between mitosis and meiosis? In mitosis, in the process of mitosis, cells are multiplying, and in process of meiosis, reduction division is taking place. Cells are reducing and here they are multiplying. Okay. So according to concept of mitosis, multiplication, and here in the first phase, that is multiplication phase, this primordial germ cells, we have seen that they are the origin of formation of sperms. From this primordial germ cells, the sperms are forming, and these primordial germ cells, what now they are going through? They are dividing more times. They are dividing several times. But by what process they are getting divided? They are getting divided by the process of mitosis. Why they are not getting divided by the process of meiosis? Because here they have to multiply. They don't have to reduce. And if they have to multiply, they have to undergo the process of mitosis and mitosis only. And here, these primordial germ cells, they will divide, and this chain or this cycle will goes on. And what they will produce after division, primordial germ cells, after these are the primordial, we can say primordial germ cells, and these primordial germ cells, they will divide, and what they will form now? Spermatogonia. And the second thing which you have to keep in mind is that when the process of mitosis is going on, see, mitosis will occur in the somatic cells, meiosis will occur in the germ cells, but this process of mitosis, multiplication is going on, and when the cells are in the process of mitosis, they are in diploid condition. Okay, they are in diploid conditions. They have two sets of chromosome here. That means they have 46 pairs of chromosomes. They have 46 pairs of chromosomes present. And these primordial germ cells, they will undergo division by the process of mitosis, and now they are getting multiplied. But whenever they are getting from this one primordial germ cell, two cells are forming, but their genetic material is not getting divided. They are just multiplying, and here they are having a 2n condition, and after multiplication also they are showing 2n condition only. But here we are calling it as a spermatogonium. Okay, spermatogonium A and spermatogonium B. Now what is the difference between spermatogonium A and B? There is not much more difference between spermatogonium A and B. Just it is the first step of division, and after first step, the second step is known as spermatogonium B. That is only difference. There is not any difference in chromosomal material. Both of them are having 2n condition only. Got it? Now, in the next step, in the growth phase, in multiplication phase, what we have seen? We have seen that cells, primordial germ cells, are getting divided. Not divided, they are getting multiplied. And when they are getting multiplied, they are not undergoing reduction division, but they have same 2n condition as the primordial germ cells are having. Now in this growth phase, what happens? Multiplication, the process of multiplication is not seen here. Here only the growth of that cells is taking place. Now here the role is of primary spermatocyte. The primary spermatocyte will grow and develop. Why we are calling it as a primary spermatocyte? Because it has undergone the process of multiplication, and after doing the process of multiplication, it has came to this stage, and that's why it is known as primary spermatocyte. Now it is near to become the sperm. And now what happens in the growth phase? The spermatogonium, which we have seen spermatogonium. This spermatogonium, now that spermatogonium increases in the size. Here multiplication or division is not taking place, just the growth is going on. They are just growing. They are increasing in their size. Now how they are increasing in their size? Because of accumulation of nutrition or nutrients. And where that nutrients are getting accumulated? That nutrients are getting accumulated into the cytoplasm, of that cell. Again I will tell you, growth phase. What is happening in this growth phase? In this growth phase, the primary spermatocyte has a main role. Why we are calling it as a primary spermatocytes? That we will see in the next point. Now here, growth phase. First of all, what happens here? In the last slide, in the multiplication phase, what we have seen? The formation of spermatogonium, and both the spermatogonium, they have 2n condition. And now in the growth phase, these spermatogoniums are not going to divide or multiply. They are just going to increase in their size. And why they are increasing in size? Because of accumulation of nutrients inside their cytoplasm, inside their cell cytoplasm. And now after that, after getting increased in size, after accumulating all the required nutrients, what they are going to do now? Now they are getting ready for the meiotic division. For what process they are getting ready now? They are getting ready for the meiotic division. Now what is happening in this meiotic division? Now in meiotic division, reduction will take place. In the mitosis we have seen that only multiplication was going on. Here in meiosis, reduction will take place. Now what happens? From 2n condition, it will come to the n condition. And after meiotic division, the spermatocytes are known as primary spermatocytes. They are known as primary spermatocytes. Got it? This I will clear you in the next slide. Got it? Now, in the maturation phase, what happens here? In the maturation phase, now each of the primary spermatocyte, it will undergo 2 maturation division. What it will undergo? See here, it is the primary spermatocyte, and this primary spermatocyte will undergo 2 maturation division. It will again get divided into this condition, and again they will get divided into. That means here they are undergoing 2 maturation divisions. Now what is happening in first maturation division? We are calling this first maturation division as a reduction division. We are calling it as a reduction, means reducing. Now in what thing they are reducing? They are reducing copies of chromosome or sets of chromosome, and when they are reducing, see here, up to primary spermatocytes, what they are having? They are having 46 chromosomes. That means they have 2n condition. But after that, here this primary spermatocyte will undergo 2 maturation division, and when it will undergo the first maturation division, before it has 2n condition, and after that it will undergo reduction division by the process of meiosis, and now they will have 2 haploid cells. This nucleus will have N condition, and this nucleus will have, here there are 46 chromosomes, not pairs, 46, mistakenly I said 46 pairs, but there are not 46 pairs, only 46 chromosomes are there. 46 chromosomes are here, that means 23 pairs, and here after first maturation division, this primary spermatocyte, which is in 2n condition, it will undergo reduction division, or it will reduce, and it will form 2 only n condition. Now here the 46 chromosomes will get divided into 23 in one cell and 23 in other cell. In this way the reduction has taken place. Now after first maturation division, the reduction is going on and they are forming 2 haploid daughter nucleus. See here, first and second, 2 haploid daughter nucleus are formed here having N condition. Haploid means N. They are showing N condition. Both of them are haploid, having 23 number of chromosomes, and we are calling them as a daughter cells of this primary spermatocytes. And after that, when they are forming these 2 haploid daughter cells, we are calling them as, these are now after first division, we are calling it as a secondary spermatocytes. These are the secondary spermatocytes, and this secondary spermatocytes, they have 23 number of chromosomes present inside it. Now both the secondary spermatocytes 1 and 2 again undergo second maturation division. See, this will undergo second maturation division, and again this will undergo second maturation division. Again they are getting divided into, or they are forming 2 daughter cells. And at this stage, what happens? After secondary spermatocytes has undergone second maturation division, they will form 4 haploid spermatids. See now, first of all, we are saying it as a spermatogonia. After that, in the very first, when the initial stage, we are calling it as a primordial germ cells. After that, spermatogonia. After that, primary spermatocyte. And after that, haploid spermatids. How the name differ according to these stages? See, first of all, what we have said it, primordial germ cells. Wait a minute. First we said as a primordial germ cells. After that, we said these cells as a spermatogonia. And at the next stage, what we are saying it as a primary spermatocyte. Primary spermatocyte. And at the last, after reduction division, we are saying these cells as a spermatids. Got it? How the difference is seen in their names. And now, after formation of this haploid spermatids, you got, see here, 1, 2, 3 and 4. These 4 haploid spermatids, again they are N condition, again they are having N conditions. These 4 haploid spermatids are formed by or after second maturation division. After second, this was the first maturation division. What was this? This was the first maturation division. After that comes here second maturation division. Second maturation division. After first maturation division, what happens? Only 2 daughter cells are formed, and after second maturation division, 4 daughter cells, 4 haploid daughter cells are formed. In this way, the process of maturation is going on. Got it? Now, see here, whatever things we have seen in the 3 phases of spermatogenesis, they are represented in a diagrammatic form here. How? Here the types of divisions are told. Here the phases, or we can say it as a types of division, and these are the types of germ cells. Now see, first of all, and where the process is going on. This process of spermatogenesis is going on near to the testes inside the germinal epithelium cell. And in this germinal epithelium cell, first initial stage, this was the primordial germ cell. This was the primordial germ cells, and after that the primordial germ cell gets converted or they are getting multiplied, and at that stage they are undergoing mitotic division. And in this multiplication phase, what they are undergoing? They are undergoing mitosis. And with the help of this mitosis, these cells are getting multiplied. And after this multiplication here, so many spermatogonia are formed. After primordial germ cells, spermatogonia are formed, and after that, see, first primordial germ cells, second was spermatogonia, and third was primary spermatocytes. And this after going through the process of mitosis, they will form a spermatogonia, and this spermatogonia, they are undergoing some process, and we are calling it as a primary spermatocytes. And after that, this primary spermatocytes, they are undergoing the process of meiosis first, and after the process of meiosis first, they are forming secondary spermatocytes. Here was 2n condition, and here they are showing n condition. Only one set is present. Here 46 chromosomes are present, here 23 chromosome and 23 chromosome. That's why they are showing n condition, haploid condition. After that, after going through meiosis first, they will again go through meiosis second, and again this one daughter cell will again gets divided into, see, here the process of multiplication was going on, and here the process of division is going on. That's why here you are getting haploid cells, and here every time, each and every time you are getting only diploid cells. Here after second meiosis division, again many numbers of spermatids are getting. Now we are calling them as a spermatids. Got it? And after formation of the spermatids, they will form a spermatozoa in the maturation stage. But to form the spermatozoa, they have to undergo again one process. It is known as Spermiogenesis. And these are the stages of spermatogenesis. Now, which hormones are supporting during the process of spermatogenesis is going on? When that process is going on, there are some hormones which are supporting to this process. Which are that hormones? Now, at the time of spermatogenesis, or the spermatogenesis will start at a specific time, and we are calling that specific time as an age, and it has a medical term. It is known as puberty. The process of spermatogenesis starts when a organism reaches to the age of puberty, and when the organism reaches to the age of puberty, they will start the process of spermatogenesis. And because of starting the process of spermatogenesis, what happens with them? They are able to increase secretion of gonadotropin releasing hormone. Now what happens? When the age of puberty comes, that time only the process of spermatogenesis is starting, and with that the hormonal secretion is also starting. And when the start of spermatogenesis is seen, at that time only the secretion of gonadotropin releasing hormone GnRH is taking place. And from where it is secreted? It is secreted from the hypothalamus, and now that GnRH is helping for the process of spermatogenesis. See, the process of spermatogenesis as well as secretion of gonadotropin releasing hormone, both are taking place at the age of puberty only. And that's why they are related to each other. And now, when the level of gonadotropin increases, when the level of gonadotropin increases in the blood or in the blood stream, what happens? They will stimulate. To whom they are stimulating? They will stimulate the anterior pituitary gland. And after stimulating anterior pituitary gland, this anterior pituitary gland will secrete one important hormone. That hormone is known as luteinizing hormone. See, here you have seen two types of hormone. First is gonadotropin releasing hormone and second is luteinizing hormone. But both of them are having different roles. See, this GnRH, that is gonadotropin releasing hormone, it is released from the hypothalamus, and luteinizing hormone, it is released from the anterior pituitary. Okay, and hypothalamus or pituitary gland, we are calling it as a master endocrine gland, and they are secreting both of these hormones. And but now here what happens? First of all, when the level of GnRH increases in the bloodstream, then and then only the stimulus is given to pituitary gland to increase or to secrete the luteinizing hormone. And with that luteinizing hormone, again second hormone is secreted, that is luteinizing hormone and follicle stimulating hormones, FSH. And both of these LH and FSH are helping for the formation of sperm, for the process of spermatogenesis. Means what? The main role during the formation of spermatogenesis is of luteinizing hormone and follicle stimulating hormone, but they are stimulated by which hormone? Gonadotropin releasing hormone. That means gonadotropin releasing hormone has an indirect role They are just doing a role of stimulating anterior pituitary. And if they are stimulating anterior pituitary, then and then only the pituitary gland will able to secrete luteinizing hormone and follicle stimulating hormone. And when both of these hormones are secreted, that hormones will help for the process of spermatogenesis. Now, what type of help they are doing? This LH, luteinizing hormone, it will act as a Leydig's cell. It is again important for your CET and NET. LH, luteinizing hormone, they are acting at the Leydig cells, and after acting on the Leydig cells, they are stimulating. To whom they are stimulating? They are stimulating synthesis and secretion of androgens. Androgen is a male hormone. They are secreting this, they are synthesizing also, and they are secreting also the male hormone, that is androgen. And this androgen stimulates the process of spermatogenesis. See now how again the hormones are coming one by one, and how they are related to each other. First, which hormone came? Gonadotropin releasing hormone came. It was secreted. See again, here we will see hypothalamus. Okay, it is secreting GnRH, and this GnRH, if it will increase in the blood stream, what happens when this hormone increase in blood stream? It will stimulate pituitary, anterior pituitary means adenohypophysis. It will stimulate anterior pituitary gland to secrete two hormones. One is LH and second is FSH. And now this LH acts at the Leydig cells. This LH will act at the Leydig cell, and what they are doing? They are secreting androgen. They are secreting androgen, and this androgen is in turn helping the process of spermatogenesis. Now, what will be here? We have seen the role of LH. Here we have to see the role of FSH. Now, what FSH is doing? It is acting on the Sertoli cell. See, there is difference of action. LH is acting on Leydig cells and FSH is acting on Sertoli cells. Here, when FSH acts on Sertoli cells, again it will secrete something. It will secrete two factors. Which are those two factors? Here, first one is androgen binding protein. This androgen which is secreted by LH. And that androgen binding protein is secreted by follicle stimulating hormone. And with that, inhibin is also secreted. And this is helping the process of spermiogenesis. See, spermatogenesis. Here, LH is supporting to the process of spermatogenesis and FSH is helping to the process of spermiogenesis. And there is difference between spermatogenesis and spermiogenesis. Now see, we will discuss the process of spermiogenesis now. Before that, we have discussed the process of spermatogenesis. What was spermatogenesis? That we have discussed. There it is having three phases: multiplication, growth and maturation. But now here we are going to discuss spermiogenesis. Now what we have seen, that it is a process of transformation of spermatids. It is the process of transformation of spermatids to spermatozoa. In the last slide, see, I told you primordial germ cells, they are getting into spermatogonia, after that primary spermatocytes, and after that spermatids. And here, after formation of spermatids, they have to form into, or they have to transfer into spermatozoa. But to spermatids to go in the form of spermatozoa, they have to undergo the process of spermiogenesis. With the help of process of spermiogenesis, they are converting from spermatids to spermatozoa. And up to this stage of spermatids, they are not able to travel. But here, when they will get converted into spermatozoa, now we can call them as a sperms, because here they are having 10. The spermatozoa and this spermatozoa, they are known as sperms. And these spermatids becomes, again, the spermatids, what happens? These spermatids, if they have to become spermatozoa, they have to have a part, that is head, Golgi apparatus, and that Golgi apparatus should have proteolytic enzymes and acrosome cap. When the sperm has all these things present in it, we can call it as a spermatozoa. Now there are again different phases of spermiogenesis. Now which are these different phases of spermiogenesis? It is again having 4 phases. First one is Golgi phase, second one is cap phase, third one is Tel phase, and fourth one is maturation phase. And in this phase, what happens here? Spermiogenesis, formation of sperm. It is again known as spermiation, means again formation of spermatozoa. The spermatozoa, from where they are released? They are released from the seminiferous tubules. And after releasing from the seminiferous tubules, because in the lining of seminiferous tubules only, they are having a lining of germinal epithelium cell, and in that germinal epithelium cell only primordial cells are present, and again in between that nerve cells are present. We are calling it as a Sertoli cells. And when the spermatozoa are released from seminiferous tubule, now they have to migrate to different place. They are migrating towards the epididymis, where they become fully differentiated motile spermatozoa. And in this step, here when they migrate to the epididymis, and here they will become fully differentiated motile spermatozoa, and here we can say them as a sperm. After completing the process of spermiogenesis, we can say them as a sperm, because now they have head region as well as they have Golgi phases, they have Golgi present, they have tail, they have cap. All these things are present in the sperm or the cell, and that's why we are calling it as a spermatozoa, and that's why they are able to motile. And if what happens here, the sperms are motile, and if there are any disturbances in the process of spermiogenesis during formation of sperm, if this spermiogenesis is disturbed, that time what happens? It will give rise to sperm cells with abnormal morphology. Means maybe when मतलब स्पर्मियोजेनेसिस में अगर प्रॉपर डेवलपमेंट नहीं हुआ, ये फेजेस में हर एक फेज सीरियली नहीं गया, उसके अंदर प्रॉपर स्पर्मियोजेनेसिस का प्रोसेस नहीं हुआ, तो क्या हो सकता है? कुछ डिस्टर्बेंसेस इसमें अगर आए तो क्या होगा? दे विल क्रिएट एबनॉर्मल मॉर्फोलॉजी, मीन्स अपीयर इरेग्युलर, and because of that, it will affect fertility of males. Okay. That's why the process of spermiogenesis is also important for the sperms, for the formation of sperms. It is the important process. Now see here, this is the structure of sperm. Here the head of sperm is present. Here the midpiece is present, and this is the tail region, and this is the end piece. And here inside the head region, nucleus is present, and at the starting or at the ending of head region and starting of midpiece, what is present? Centriole is present, and again a mitochondria is present in this midpiece. And what you have seen during the process of formation of zygote, what has happened? During the process of formation of zygote, what happens? The head is only entering inside the egg, and that head is having only nucleus, but the mitochondria remains outside the cell, outside the egg. That's why we people, we all have a mitochondria. We have a maternal mitochondrial origin. Whatever mitochondria is present in our cell, it is from our mother. It is not coming from our father. It is coming from our mother only. Okay, got it? Now, this sperm, again, it is having acrosome. It is the covering where the acrosome is seen. And now what will be the role of this acrosome? We will see here. Now, one by one, we will see first of all the phases of spermiogenesis. In the Golgi phase, what is going to happen now? In the Golgi phase, we are calling this phase as a initial event of spermiogenesis. It is the starting of spermiogenesis. And here, spermatids, up till we are not able to call these spermatids as a spermatozoa. That's why we are calling it as a spermatids. These spermatids, they are prepared. For what purpose they are prepared in this Golgi phase? These spermatids are prepared to acquire the major parts of sperm cells. Here in this Golgi phase, the spermatids are going to acquire major parts of sperm cell. That is, which are the major parts of sperm cells? Head, midpiece and tail. See here. Here we have seen head, midpiece and tail region. But before undergoing the process of spermiogenesis, we are calling it as a spermatids only. And when they are in a spermatid stage, that spermatids are not divided into all the structures. But when the spermatids undergo the process of spermiogenesis, they will get divided into major parts of sperm cells. What are that parts? Head, midpiece and tail region. Now, what the Golgi apparatus is showing here? Here, the Golgi apparatus is showing intensified activity, and during this phase again, what is happening? The organelles, that Golgi apparatus, it produces and releases lytic enzyme. This Golgi, which is present inside the head region. See, inside the head region, Golgi is present. This sperm, it is one type of cell only. That's why whatever things the normal cell is having, just like us that, the sperm cells is also having. Other cells, normal cell have different organelles present inside their cytoplasm. Like that only the sperm, it is having a cytoplasm. Inside that cytoplasm, nucleus is present, ER is present, Golgi is present, all these things are present here. But in the first phase, that is Golgi phase, what happens? What this in this Golgi phase, what is going to produced here? In this phase, organelles produces or releases lytic enzyme. This Golgi will produce and release lytic enzyme. Now, what will be the role of lytic enzyme, or where will be the role of lytic enzyme? When the fertilization is done, at that time, sperm have to change its structure. And for that reason, lytic enzymes are working. The head region of sperm is going to change its structure, and that change in structure is done with the help of lytic enzyme. Now, what actual lytic enzyme are doing? They will gather inside the acrosome. Acrosome means what? A cap of sperm. See here, it is the acrosome. This yellow region, it is the acrosome. We are calling it as a acrosome. And inside that acrosome, lytic enzymes are present. These lytic enzymes, now, how they are helping to the sperm? The other end of spermatids, midpiece is forming. Okay, midpiece is also forming here. And the third thing which is taking place is mitochondria. The mitochondria, in this mitochondrial region, it is getting thickened, and it is getting filled in this part. In which part? In which part mitochondria is getting filled? Mitochondria is getting filled in the midpiece region. See here, again we will go towards the diagram. Here in the midpiece, this is the midpiece, and here mitochondria is getting filled. That mitochondria is present in that region. Now, normally in the normal cell, mitochondria is present inside the head region only. We can say mitochondria is present near to the nucleus, or it is present or embedded inside the cytoplasm. But here what happens? Now, the head region of sperm do not contain mitochondria, but that mitochondria is present in the midpiece of the sperm after centriole. Okay. And again, what the next thing is going on in this phase, that is the genetic material is undergoing packaging. And where the genetic material is present now? Again, we will go towards the diagram. The genetic material is present inside the nucleus, and inside that nucleus, that genetic material is getting packed with what? With the histone protein. This genetic material is getting packed with the help of histone proteins inside this nucleus. And by the end of this phase, the genetic material is packed, it is getting highly condensed, and it is in that genetic material, the process of transcription is not going on. It is inactive transcriptionally. Okay, here what we have seen in Golgi phase, here we have seen the spermatids. The changes in spermatids are going on. The spermatids, what they are doing? They are preparing to acquire major parts of sperm cells. They are prepared. The spermatids are going to acquire major parts of sperm cells. And which are those major parts which we have seen? That are head, midpiece, tail region. And here, now what happens in the head region? In the head region, the activity of Golgi apparatus is seen. Here, in the head region, it is head region which is covered by a acrosome. Head region is covered by acrosome, and inside that acrosome, we are calling it as a acrosomal cap, and that cap contains, it is filled by lytic enzymes. And after that, first thing, which organelle is formed? Head, and above the head region, Golgi apparatus is seen, and it is filled by lytic enzyme, and we are calling it as an acrosome. Second thing what we have seen, again the midpiece is getting developed. At the other end, midpiece is getting developed, and this midpiece, when it is getting developed, means what is happening with this midpiece? In this midpiece, that area becomes thickened. And why it is getting thickened? Because in that area is incorporated by the mitochondria. Normally, mitochondria is present inside the cytoplasm, but now it had come to the midpiece region. And the third thing is, during this phase, inside the nucleus, the genetic material is undergoing packaging with the help of histone proteins. And the fourth thing was that they are transcriptionally inactive. The process of transcription is not going on inside this cell now. Got it? These four things are happening here. Now, next phase, after Golgi phase, the second phase is cap phase. This cap phase means what? This is the phase which is characterized by events leading to formation of acrosomal cap. Here, suppose this is the sperm, and here it is forming a cap region. We are calling it as a acrosome, and inside that acrosome, lytic enzyme is present. Lytic enzyme is present, and this is the acrosomal cap region. Now what happens in this acrosomal cap region? This acrosomal cap, or we can call it as an acrosome also, and it is a membrane bound compartment. See here, this is the sperm, okay, and above that sperm one membrane is present, and inside, in between sperm membrane and this acrosomal cap membrane, what is present? Lytic enzymes are present, and where they are present, they are located at the tip of sperm, at the tip of sperm head, and it forms when Golgi apparatus surrounds the anterior of spermatids to form the acrosome. Normally, we don't have to study hard in this language. Normally, you can say that this is the sperm region, this is the membrane of sperm, and above that sperm, above the tip region of head of sperm, what is present? It is covered by the acrosomal cap, and inside that cap, what is present? Lytic enzymes are present, and it is made up of Golgi apparatus. This only you have to study, and here you have completed with cap phase. Now next phase is Tel phase. In the Tel phase, what happens? How the Tel phase is characterized? How can we say that now the process of spermiogenesis is in the Tel phase? Now it is characterized by elongation of microtubules on one of the centriole of spermatid. Now also we are calling it as a spermatid. See now. Here we have seen it is a centriole present, and this centriole, this is the centriole, and now it will form a tubular structure, and here so many microtubules are getting added. Microtubules are adding again and again, one by one, they are always adding, and when they are added, they are increasing. The length of tail is getting increased. Got it? And the length of tail is getting increased with the help of elongation of microtubules on the centrioles. Now the microtubules, how are microtubules? Microtubules form an axoneme, and this axoneme contains microtubules that are arranged in the 9+2 configuration. See here, see, this is the if we will say that this is the centriole, and from this centriole, what happens? These microtubules are getting added to this. And how will be the structure of this centriole? This is the structure of centriole. It is showing 9+2 arrangement. See, 1 and 2. Okay. And how 9? See, 1, 2, 3, 4, 5, 6, 7, 8 and 9. That's why we are saying it is showing 9+2 arrangement. These are the 9 different, what we can say them as they are subfiber A and subfiber B, and inside they are showing again two complete subfibers, and here one is subfiber A which is complete and subfiber B is incomplete. Any one fiber is incomplete here, and in this way they are forming 9+2 arrangement. Now why they are doing this 9+2 arrangement? Here when this centriolar region, and here in the centriolar region one by one microtubules are getting arranged, and when microtubules are getting arranged they are forming a tail region, and because of that arrangement the tail will become flexible, and now the tail is able to locomote. Because of arrangement of these microtubules, the tail will locomote. Otherwise, the tail will not be able to locomote. Because of this only, because of this, the tail is able to locomote because of axoneme structure, and this axoneme, it contains microtubules, and because of that it is able to dilate, or it is able to locomote in the female reproductive tract. Okay, now the last phase of spermiogenesis is maturation phase. Now what will happen in this maturation phase? It is the final phase of spermiogenesis. Here the cytoplasm, which is a part of sperm cell, as well as the remaining cytoplasm and remaining organelles. Which are the remaining organelles? Here we have seen the Golgi apparatus, we have seen nucleus, we have seen centriole, we have seen mitochondria. But remaining organelles, other than this, they are removed. Remaining cytoplasm as well as remaining organelles, they are removed by the phagocytosis process. And now what will be the essential role of Sertoli cells in spermatogenesis? We have seen in spermatogenesis that Sertoli cells, they are the nurse cells. Here in maturation phase, only this thing is taking place, that remaining cytoplasm and organelles are removed by the process of phagocytosis. Now this is the next subpoint. What will be the essential role of Sertoli cells in spermatogenesis? Now here, up to this, we have completed with Spermiogenesis. This was the last phase of Spermiogenesis, maturation phase. Now, after removing all the remaining cytoplasm and organelles of the sperm cell, we can call it as a spermatozoa now. Now, spermatozoa has formed. Now, we can't call it as a spermatid. Now, we can call it as a spermatozoa because it is motile now. When it was in a spermatid stage, it was not able to locomote. It was not motile. But after getting converted into spermatozoa, it is motile. And we can call it as a sperm now. Now, the next subpoint is essential roles of Sertoli cells in spermatogenesis. How the Sertoli cells are helping in the process of spermatogenesis. What they are providing? They are providing nourishment to the germ cells which are attached to them. Okay. And They are also responsible for secretion of testis determining factor, and because of that they concentrates testosterone. Again, then what they are doing? Then in spermiogenesis, how they are helping? In spermiogenesis process, they are responsible for phagocytosis of residual cytoplasm. This process only. See here, these Sertoli cells, what they are doing? They are only helping in the maturation phase of spermiogenesis to remove cytoplasm and other organelles by the process of phagocytosis. And the final product of spermiogenesis is non-motile. Here I am saying it as a non-motile mature spermatozoa. But when will it become motile? Now it will become motile when, see here, the spermatozoa after this stage will still be sterile since they are not yet motile. Now they are not yet motile, but when they will become motile? They will become motile cell when they further develop into epididymis, and the migration of spermatozoa to the epididymis will form a motile sperm cells, and now we are saying this process as a spermiation, and after completing the process of spermiation we can call them as a sperm, or we can call them as a motile sperm. Got it? Here we have completed with spermatogenesis, and spermiogenesis what was the part of spermatogenesis. First of all we have seen spermatogenesis, first of all we have seen gametogenesis, and before that we have seen what we have seen? Different steps in human reproduction. One by one we have discussed those steps, and from that we have started with first step of human reproduction, that was gametogenesis, and we all know that there are two gametes, female gamete and male gamete. Today we have completed with formation of male gamete, and formation of male gamete, that process is known as spermatogenesis, and that spermatogenesis again have three steps: spermatogenesis, spermiogenesis and spermiation. And after completing these three steps, that sperms are becoming motile and we are able to call them as a sperm, and now they are active in their function. Got it? So today we have completed this much syllabus from our 12th biology, and after that we will meet in our next lecture with different topic from 12th biology only. Up to that, take care of yourself. Bye.